The gentle, rhythmic flashing of a firefly in an open field feels like magic, but the biological machinery generating that light is so precise and efficient that it has become a foundational tool for global biowarfare defense and environmental monitoring. For centuries, human culture has viewed bioluminescence as a symbol of pastoral serenity. We see floating sparks of cold light and associate them with ephemeral beauty and peaceful summer evenings. That harmless reputation took a strange detour during the Cold War. In the 1960s, a classified U.S. military operation called Project Firefly launched hundreds of sounding rockets into the upper atmosphere to test early warning missile sensors. Because the project shared a name with the beetle, it fueled a persistent myth that the military was weaponizing glowing insects, even though the project was entirely unrelated to biology. Both our romanticized folklore and our Cold War conspiracies obscure a much darker reality. The truth about why these beetles glow requires us to look at an ancient, highly evolved biological war for survival. Producing light requires a massive expenditure of cellular energy. In a competitive ecosystem, wasting energy is a death sentence. Terrestrial bioluminescence did not originally evolve so insects could find mates. It evolved as aposomaticism, a stark biological warning signal to predators. Long before they grow wings, firefly larvae emit a steady green glow on the forest floor. That light advertises the presence of lucibophagens, which are heart-stopping toxins. If a bird or a lizard eats a glowing larva, the steroids can be lethal. Over millions of years, fireflies transitioned to nocturnal activity and repurposed that defensive glow. The steady warning signal of the larvae evolved into the complex synchronous flashing language used by flying adults for reproductive selection. This light is a programmable biological alarm system, forged over millions of years of evolutionary warfare. To understand how humans co-opted this alarm system, we must look inside the beetle's abdomen. The firefly produces cold light using a precise chemical engine. It combines a substrate called D-luciferin, cellular ATP, and molecular oxygen, catalyzed by luciferase. The enzyme creates an unstable four-membered ring. A molecular cleavage snaps the oxygen bond apart, and as electrons drop, excess energy is emitted as a single photon of visible light. For decades, scientific literature perpetuated the belief that this reaction was perfectly efficient, losing zero energy as heat. Modern computational chemistry proves the actual quantum yield is roughly 41%. The insect achieves its intense perceived brightness mechanically. The exoskeleton over the lantern is covered in microscopic, jagged, asymmetric scales. These structures prevent light from reflecting backward into the body, extracting 90% of the generated photons into the environment. Once chemists decoded this physical and biochemical architecture, they realized they could isolate the engine entirely. By extracting the biological machinery, scientists gained the ability to plug a living light source directly into modern technology. In medical oncology, researchers use bioluminescence imaging to track cancer. By genetically tagging cancer cells with luciferase, tumors quite literally light up as they metastasize inside living animal models. This allows scientists to visually monitor disease progression and the impact of chemotherapy in real time. The military applications have evolved just as drastically. During World War I, soldiers gathered European glowworms in glass jars. The faint bioluminescence was bright enough to read maps, but dim enough to avoid drawing deadly sniper fire. Today, the focus has shifted to rapid threat detection. Defense agencies now use engineered reporter phages to actively hunt down weaponized Category A pathogens, like anthrax and the plague. Scientists have spliced the LuxAB bioluminescence genes into these phages. When they locate a lethal pathogen in a blood or environmental sample, the virus hijacks the target's cellular machinery and forces the bioweapon to rapidly manufacture light. This provides a critical operational advantage. Traditional laboratory cultures for these pathogens require up to 36 hours. The reporter phage triggers optical alarms in under 60 minutes, providing real-time situational awareness during an active bioweapon release. The same system can detect synthetic chemical weapons. Extremely lethal nerve agents like sarin and VX actively inhibit the luciferase enzyme. Military sensors can detect these neurotoxins at parts per billion levels, simply by measuring the dimming of the bioluminescence. The chemical alarm system that originally warned predators away from a toxic beetle has become an engineered defense grid, warning humanity of invisible nerve agents and weaponized plagues. Bioluminescence is also changing how we build our cities through biomimicry. Engineers are studying the microscopic structure of the firefly to solve major physics problems in commercial lighting. In standard LEDs, much of the generated light reflects off internal boundaries and is lost as heat. By 3D printing the jagged, asymmetrical scales of the firefly onto synthetic LEDs, engineers broke that internal reflection, boosting light extraction efficiency to 90%. Researchers at MIT are already developing treatments that could transform mature trees into self-sustaining light sources using their own photosynthetic energy. This has already moved from the lab to the consumer market. The firefly petunia is a genetically modified plant that glows continuously by utilizing a fungal pathway. It converts a common plant molecule into a light-emitting substrate, allowing the plant to shine throughout its life cycle. Bioluminescent technology is moving into the realm of infrastructure, offering a path toward lighting our cities without relying on electrical grids. Writing light permanently into the DNA of consumer plants creates a massive ethical dilemma. As we turn living organisms into engineered machines, bioethicists face complex challenges regarding containment and oversight. The unregulated release of these organisms poses significant ecological risks. If these novel genetic traits transfer to wild relatives, the resulting light pollution could disrupt local food webs and impact the behavior of light-sensitive species. There is a tragic irony in this development. Human light pollution and habitat loss are already threatening the survival of wild fireflies. Our attempt to commodify synthetic light could further stress the very species that inspired the technology. Nature's ancient survival mechanisms have illuminated the hidden mechanics of human disease and fortified our modern military defenses. Having adopted its light to secure our cities and track our diseases, we must now ensure that our pursuit of the future does not extinguish the natural wonders that guided us there.